Hello friends, welcome to Bioassembly. In this session, I will discuss about lysosome and their structure, types and function. Let's get start the session. First of all, lysosome was discovered in 1955 by C.D. Dewey and named this organelle as lysosome. Lysosome is organelle which is provided an excellent example of ability of intracellular membrane to form closed compartment in which compositions of the lumen aqueous interior compartment differ substantially from that of the surrounding cytosols. Means lysosome provide excellent example of intracellular membrane and it closed compartment, it composite of lumen and differ from substantially surrounding cytosol. Found exclusively in animal cell, lysosome are responsible for degrading certain components that have become obsolete for the cell organism. Means lysosome degrades certain types of component that is not necessary for the cell organism. Lysosome are often buried from the membrane of Golgi apparatus, but in some cases they develop generally from the late endosomes, which are vesicles that carry material brought into the cell by process known as endocytosis. Means lysosome develop in some cases from the late endosome and which are vesicles. Late endosomes are vesicle and which carry materials that brought the cell by the process of endocytosis. The biogenesis of lysosome required the synthesis of specialized lysosomal hydrolase and membrane protein. Means synthesis of lysosome required the synthesis of specialized lysosomal hydrolase and membrane protein. Both classes of protein are synthesized in endoplasmic reticulum and transport through Golgi apparatus and then transported from the trans-Golgi network to an intermediate compartment by means of transport vesicle. In previous lesson lecture, I will discuss about I am discuss about the Golgi complex and where the lysosome how lysosome formed from the mature phase or trans phase of Golgi complex. These are the structure of lysosome. You can see that the lysosome is present in the inside the cytosol and lysosome are a small vesicle. You can see that these are small vesicles which are bounded by single membrane. These are small vesicle and bounded by single membrane and it contain hydrolytic enzyme. It contain hydrolytic enzymes in the form of minute crystal or semi-crystalline granules. These are semi-crystalline granules and we, which have 5 to 8 nanometer thickness. About 50 enzymes recorded in occur in semi-crystalline granules and all the enzyme do not occur in some of lysosome but there are different sets of enzyme or different type of lysosome. The important enzyme of lysosomes are acid phosphatase, protease, peptidase, nuclease, lipase and carbohydrates. These are the important enzymes present the lysosome and so lots of enzymes present inside lysosome, so it is called acid hydrolase. This all about the structure of lysosome. These are the formation of lysosome. How lysosome formed? First of all, lysosome believed to be formed from the joint of endoplasmic reticulum. These are rough endoplasmic reticulum and smooth endoplasmic reticulum. And jointly activity of endoplasmic reticulum. Lysosome formed from jointly activity of endoplasmic reticulum. or endosome. And these are Golgi complex jointly to form lysosome, and the precursor of hydrolytic enzyme mostly synthesized at the rough endoplasmic reticulum. Means the hydrolytic enzyme mostly synthesized in the rough endoplasmic reticulum, and later it transferred to smooth endoplasmic reticulum and to forming Golgi, and it formed Golgi phase or Golgi complex directly from smooth, and then this is transferred to Golgi apparatus or Golgi complex from the smooth endoplasmic reticulum. And inside the Golgi complex, the precursor are changed into enzyme, and these precursor are changed into enzymes, and the enzymes are then packed in larger vesicles which are pinched off from the maturing phase. Means the Golgi complex, I am also told about the Golgi complex. This contain two phase, cystrina, cis phase or trans phase. Trans phase is called maturing phase, and the precursor of the enzymes are formed from the trans phase. Golgian vesicles are joined by endosome to provide lysosome, and these are the Golgian vesicles joined by endosome, and it provide lysosome. Lysosome are passed through various stages in the same cell. Means these are the cell, and lysosome pass through these various stages. And depending upon their morphological structure, lysosomes are of four types. First, primary lysosome, and secondary lysosome, residual body, and autophagic vacuole. These are autophagic vesicles. See their structure. And first of all, primary lysosome. Primary lysosome when newly pinched off vesicles from the Golgi apparatus. Means primary lysosome come from the Golgi apparatus, which generally fuse with some endosome to become fully functional, and it fuse with some endosomes, and it become fully functional. Primary lysosome are These are primary lysosome, and these are small contain hydrolytic enzyme in the form of granules. These are granules, and it contains small hydrolytic enzyme, and when it forms secondary lysosome. Primary lysosome further forms secondary lysosome, and secondary lysosome is also called heterophagosome or digestive vacuoles. A secondary lysosome is formed by the fusion of food containing, means these are the phagocytosis when solid food particles enter into the cell, and these food vacuoles or phagosomes, and this phagosome or food vacuoles fuse with the secondary lysosome, primary lysosome, and it forms secondary lysosome. And here the food particle digestion occur. Inside the secondary lysosome, food particles are digested, and digested food passes into the cytoplasm. These are the digested foods passes cytoplasm, and finally secondary lysosome left with undigested food. And these are the secondary lysosome and left with undigested foods, and these are undigested food. Oh, and it forms secondary lysosome. And next this it forms secondary lysosome further form residual body. You can see that residual body. They residual body are also called tertiary lysosome. They are those lysosome which only indigestible food material have been left. Means residual body contain indigestible food material. The residual body or lysosome pass outwardly. These are passes outwardly and fuse with plasma membrane, and this fuse with plasma membrane, and through the debris into the external environment or exocytosis or efg. And these are autophagic vacuoles. Autophagic vacuoles or autolysosome. They are produce the fusion of number of primary lysosome. When number of primary lysosomes are fused, and these are from autophagic vacuole. The they are produced by the fusion of primary lysosome and around the degeneration intracellular organelle. The cell debris are digested in the autophagic vesicles or autophagic vacuole, and these lysosomes are also called disposal bag because all the cell debris are digested inside the autophagic vacuole is also called disposal bags. The digested products are made available to the cell for new synthesis, and lysosomes are therefore known as recycling center. Means digested products are available for the newer synthesis, and so the lysosomes are also called recycling center. This all about the structure and types of lysosome. And the term autolysis is the self-destruction of cell, tissue or organ with the help of lysosome. Lysosome performing autolysis due to enclose of structure to broken down instead of themselves burst or release a digestive enzyme. When lysosome are autolysis, when the cell present in the inside the lysosome are digested, and it cell are burst, and this process and it release all the digestive enzymes, and this process is called autolysis. Autolysis generally occur when the cell aging of cells, dead or diseased cell. This all about the formation, how formed lysosome, and and type of lysosome. Occurrence: the lysosome occur in most animal and few plant cell. They are evident in bacteria and mature mammalian erythrocyte. Few lysosomes occur in muscle cell and acinar cell of pancreas. Leukocyte, especially granulocytes, are particularly rich source of lysosome. Means lysosome are absent in bacteria and mature mammalian erythrocyte, and few lysosomes occur in muscle cell and pancreas cell. Particularly, granulocytes are the rich source of lysosome. Their lysosomes are so large in size they can observe under the light microscope. They are also numerous in epithelial cell of obstructed excretory organ, intestine, liver, and kidney. They occur in abundance in epithelial cell of lungs and uterus. Phagocytic cells and cells of reticuloidal system, bone marrow, spleen, and liver are also rich in lysosome. Means they are mainly present in epithelial cell, lungs of uterus, phagocytic cell, cell of reticular endothelial system, bone marrow, spleen, and liver. Structure: lysosome are round vacuolar structure bounded by single unit membrane. Their shape and density are vary greatly, and 0.2 to 5 micrometer in size. Since size and shape of lysosome vary from cell to cell and time to time, their identification become difficult. Means lysosome is difficult to identify. Lysosomal membrane: the lysosome membrane is slightly thicker and that of mitochondria, that it contains substantial amount of carbohydrate material, particularly sialic acid. In fact, most lysosomes membrane protein are usually highly glycosylated, which may help to protect them from lysosomal proteases in the lumen. The lysosomal membrane has another unique property of fusing with other membrane of cell. Means lysosome membrane are thicker, and It contains many amounts of carbohydrate material, salic acid, and lysosomal membrane also contain protein in highly glycosylated form, and which protein help to protect them and lysosomal protease in the lumen. Lysosomal membrane, another unique property is to fusing with other membrane cell, means lysosome, these are lysosomal membrane and these are vacuoles, and this form and it forms it form to primary lysosome, means lysosomal membrane is property unique property is to fuse with other membrane material. The property of fusion has been attributed to highly proportion of membrane lipid present in some micellar configuration. Surface active agents such as liposoluble vitamins, steroids, sex hormone, and destabilizing influence causing the release of lysosomal enzyme due to rupture of lysosomal membrane. Drug like cortisone and hydrocortisone and another tend to stabilize the lysosomal membrane, uh, have an any anti-inflammatory effect on the tissue. Means drugs like hydrocortisone and other stabilize the lysosomal membrane because it have an anti-inflammatory effect on tissue. The entire process of digestion is carried out within the lysosome, means like entire process of digestion of foods carried out within the inside the lysosome. Most lysosome enzymes act as a in acidic medium, means lysosomal enzyme act as an acidic medium. Acidification of lysosome content depend on ATP dependent proton and pump, means acidification of lysosome it depend on ATP dependent proton pump which is present in the membrane of lysosome and accumulated H plus inside the organelle. Lysosomal membrane are also contain transport protein. These are the membrane of lysosome and it contain transport protein and that allow the final product of digestion of macromolecule to escape so they can expected to reutilize by the cell. This all about the lysosomal membrane, and these are the structure of how lysosome food content digested inside the lysosome. First of all, I'm also discuss about ki this lysosomes. These are the plasma membrane. These are endoplasmic reticulum and the vesicles are transported in the Golgi apparatus, and here the transphase is formed mature and it form is also called mature phase and it form vesicles inside the id and a small vacuole is lysosomes. And this lysosome fused with when food particle come inside the cell by phagocytosis, and this food particle fuse with lysosome and it form primary lysosome, and the here the inside the primary lysosome digestion of uh, and it forms secondary lysosome, and inside the secondary lysosome digestion of food occur, and the all undigested food is released from the cell by exocytosis. And lots of lysosome is primary lysosome forms inside the Golgi complex, and these all are fused to form the autophagy vacuoles, and these autophagy when the cell are disease cell or aging, then autophagy vacuole burst and the all cells are die. This all about the structure of lysosome. Let's come to the function of lysosome. Lysosomes serve as a digestion compartment for cellular material, and they have exceeded their lifetime and otherwise no longer useful by autophagy. When cell die, lysosome membrane rupture and enzyme are liberated, and this enzyme digests the dead cell, and in this process the metamorphosis of amphibians, tunicates and many embryonic tissue. Example, gills, fins, tails are digested by lysosome and utilized by other cell. Second, lysosome break down cellular waste product. fats, carbohydrate, protein, and other macromolecule into simple compound, which are often then transferred back into cytoplasm as new building materials. To accomplish the task associated with digestion, the lysosome utilizes 40 different type of hydrolytic enzyme, all of which manufactured in the endoplasmic reticulum and modified in the Golgi apparatus. First of all, lysosome is a cellular digestion of cellular material, and this contain many digestive enzymes, and it help to digest dead cell. The process of metamorphosis, with the help of lysosome, all gills, fins, and tails are digested by the lysosome and utilized by other cell. Second, lysosome break down waste products, fats, carbohydrate, protein, and other macromolecule into simple compound, which are then transferred to the cytoplasm as a cell building material. And the lysosome utilize 40 different type of hydrolytic enzyme, all of which manufacture endoplasmic reticulum and Golgi apparatus. Third function is the digestion of large extracellular particle. Lysosome digests the foods contained of the phagosome or pinosome. Means these are the food particle and enter the inside the cell, and it form the inside the vacuoles, and the primary lysosome is fused to form secondary lysosome, and with the process and this process help to digestion of the food particles, and undigested food particle is left by the cell, and this process is called exocytosis. The lysosome of the leukocyte enable to later to devour the foreign protein and bacteria virus. Extracellular digestion. Lysosome of certain cells such as sperm and discharge and their enzymes outside the cell during the process of fertilization. Lysosome enzyme digest the limiting membrane of the ovum and panthera path in the ovum for sperm. Acid hydrolyses are released from osteoclast and break down bone of reabsorption. These cells are also secreted lactic acid, which make the local pH enough to optimal enzymic activity. Likewise, preceding the ossification, bone marrow formation, fibroblast release of cathepsin D enzyme to break down the connective tissue. This all about the function of lysosome. When we talk about extra function, more function, then it help to lysosome also help in cell division, genetic change, and lysosome defends body. When the lysosome or leukocyte devour foreign protein and toxic substance, bacteria, and other microorganisms, they thus take part in the natural defense of the body. And lysosome help to disposal of useless cell by the process of aging, when aging or dead cell. This all about the function of lysosome. So this all about the structure, types, and function of lysosome. Thank you so much. If you like this session, please like, share, and subscribe. Thank you for watching.